also, I would like to thank to the conference organizer to invite me to speak here. And so what I'm going to talk about is bicarbonate transporters and the intracellular pH regulation. And pH regulation or acid-base homeostasis, this is actually the fundamental problem in our body, and we have to maintain the fine our acid-base regulation in a very fine-tuned model. The acid-base balance can affect a series of processes in our body. For example, like cell survival, cell migration, and protein folding, and even cell signaling, bone remodeling, and neural excitabilities, and can affect muscle contraction, even and fertilization, and many more processes. So acid-base disturbance in our body can cause or even associated with a series of disorders and disease. And probably the hallmark of acid-base disturbance is renal tubular acidosis, then which is accompanied with a series of other symptoms. For example, ocular disorders like band cataract, and also central nervous system disorders like epilepsy, migraine, and mental retardation. And it's also associated with cardiovascular disease like hypertension, and also some development problem like short stature, and also dysfunction of acid-base transporters can cause infertility, and also associated with like cancers like breast cancer and so on. And so we all familiar with buffer system. When we do experiments, we have to use the buffer system in chemical reactions. We use like phosphate buffer or trace buffer, Heps buffer. However, in our body, the major buffering system is contributed by CO2 bicarbonate, which contributes for about 70% of buffering power in our body. And bicarbonate can combine protons to form carbonate, and then we dissociate into water and CO2. So in our body, the transport of acid-base related species across the membrane is very important in pH regulation. Here, like transport of bicarbonate across the cross membrane, and also proton. So it's protein transporters, and also the transport of CO2 gas molecule across the membrane. So there's the issue of gas channels. So this is basically the problems we are concerned about. And first, I will talk a little bit about the gas channel with the work I did when I was a postdoc in Dr. Warren's lab. And the conventional view about gas transport is that is the principle we call Overton rule, which was proposed by the British physiologist more than 100 years ago, Overton. He proposed that gas molecules can just move through the membrane and freely, just by simple diffusion, and no protein is required to facilitate this process. However, we found that at least some membrane proteins can facilitate enhanced membrane permeability to gas molecules. So we proposed a gas channel hypothesis. Now I will introduce briefly the technique. We use larvae oocytes as an extraction system to start the function of channels and transporters. And so we introduce the RNA encoding the proteins into the oocytes by microinjection, and then incubate the cells for expression for 4 to 5 days, and then we use electrophysiology to start the function of the membrane proteins. So let's see what will happen when we expose the oocytes into a solution containing CO2 and bicarbonate. This is the oocyte when we put the cell into a solution containing carbon dioxide. So then carbon dioxide will flux into the cell very quickly, And this process will then CO2 in the cell will combine water molecule to form proton and bicarbonate. This process will cause decrease in intracellular pH, and will cause a decrease in the concentration of CO2 around the cell, nearby the cell. So the decrease in CO2 around the cell membrane will be replenished by two mechanisms. One will be it can be replenished by CO2 in the box solution, and it can also be replenished by the bicarbonate, which will combine proton and to form CO2 and a water molecule. This process instead will cause a pH, surface pH to increase. So we use an electrode just sticking on the surface of the cell surface to monitor the changes in the surface pH. So we can see if we express a channel protein, we can see that enhanced magnitude in changes of surface pH. So this is an index of the enhancing in gas permeability. And using the similar technique, we can monitor the cell permeability and the permeability of cell membrane to ammonium molecules. And using this approach, we see that different membrane proteins, like aquaporins, aquaporin 0, aquaporin 1, aquaporin 4, aquaporin 5, and some MTB. This is the bacterial ammonium channel protein, and RHAG. This is the protein from a human. They have different. These membrane channel proteins have different permeabilities to CO2 and ammonia. Then we divide the permeability of CO2 to ammonia, then we get a relative permeabilities to different gas molecules. So this we show the gas selectivity to different gas molecules. And this is the first example of gas permeability, gas selectivity by channel proteins. And this work was done in waterborne lab when I was still a postdoc. And we did this. This project was finished by Mus Aziz and. Next, I will talk. The rest part of this talk will be about bicarbonate transporters, basically. So, acid-base transport, there are a lot of acid-base transporters in our body. And according to the effect in intracellular pH, these acid-base transporters can be classified into two different categories. One is the acid extruders. The effect of these proteins will cause increase. The activity of these proteins will cause the increase in intracellular pH. And those proteins include, for example, proton pumps. They use energy from dialysis of ATP to extrude protons from cell to extracellular. So increase the intracellular pH. And also include sodium proton exchangers, which belongs to SLC line family. And also some sodium-dependent bicarbonate transporters, for example, sodium-driven chloride bicarbonate exchangers of SLC4 family, and sodium bicarbonate co-transporters. These proteins will mediate the uptake of bicarbonate from extracellular side to intracellular. And some other proteins, these proteins we call acid loaders. They basically doing the things to extrude the base out of the cell. For the bicarbonate, basically the primarily is bicarbonate. This include bicarbonate channels and also adient exchangers. They do uptake extracellular chloride in exchange of intracellular bicarbonate, and also some sodium bicarbonate co-transporters. So these proteins will they will tend to decrease intracellular pH. And we are interested in these sodium-dependent bicarbonate transporters, which we call sodium carbonate bicarbonate transporter (NCBT). These transporters belong to the solute carrier 4 SLC4 family. This family includes 10 members, and 3 are adient exchangers. They are sodium independent, and these 5 guys are sodium dependent. The activity depends on the presence of sodium ions. These sodium NCPTs includes two different types. One is electrogenic. They transport sodium and bicarbonate in the stoichiometry, more than 1 to 2 or more than 1 to 3 stoichiometry. So the activity will affect the membrane potential and will cause hyperpolarization or depolarization. And these guys, they transport sodium and proton in 1-to-1 stoichiometry. So the process is electron neutral. They will not affect the membrane potential. If we express this transport in centimeter oocytes, and let's say what happens when we perfuse the cell with a solution containing CO2 bicarbonate. First, we expose the cell into a CO2 bicarbonate solution. This is the process we just see previously in previous slides. The CO2 fluxing into the cell will cause intracellular pH to fall. And now, if there is a bicarbonate transport expressed on the membrane, for instance, if we express NBCE1 the transporter, then this protein it will mediate uptake of sodium and bicarbonate from extracellular solution, and this process will push back the reaction to the left, and will cause a rise in intracellular pH. Okay, so first we see a decrease in intracellular pH, and then we see a very powerful increase in the intracellular pH. And this is the change of the intracellular pH is the indicator of the activity of the transporter. And also note that we also use we also see a very huge decrease in membrane potential. It's a huge hyperpolarization, because this transporter is the electrogenic transporter. It can each time it transports ions into the cell, it will bring in negative charges into the cell, so it will cause a decrease of membrane potential. This is a typical phenotype of electrogenic sodium bicarbonate co-transporters. And if we remove the extracellular sodium, then we see the transport direction is reverted. Okay, so this is the NBCE1. What if we express electroneutral NBCN1 into the oocytes? Then we can still see the huge strong pH recovery, which is an indicator of bicarbonate transporters. And however, we did not see the hyperpolarization, which we just see for NBCE1. This is a sign of electroneutral transport process. Okay. And then if we use blank oocytes without expression of transporter, then we see no recovery of pH and no change in membrane potential. Right? And so this is the topology, the structure of these transporters. It contains a huge membrane spinous domain containing just 14 transmembrane segments. So a key question we are interested in was what is the structural basis for why some transporters are electrogenic or some transporters are electroneutral. What is the structural basis to determine the electrogenicity versus electroneutrality of these transporters? So we made a series of chimeric transporters of MCU1 and MCN1, and finally we found that this extracellular loop between transmembrane segments 7 and 8 is a key structural element that can determine the structure electroneutrality versus electrogenicity. If we, by manipulation of this loop, we can convert an electrogenic transporter into an electroneutral one, and vice versa. For example, this is a white type MCE1. It is typical electrogenic. Right? We can see this huge hyperpolarization. So based on the background of this transport, we simply replace this loop with a homologous sequence of MCN1, and we abolish the electrogenicity of the transporter, but we still bicarbonate transport activity. We see the pH recovery. So, and on the other hand, this is an electroneutral chimera. This is an electroneutral chimera with the first half is MBC1 and the back half is MBCN1. This is electroneutral. So we simply replace this loop with the sequence of MBCE1. Then we get the electrogenicity of the transporter. Okay. So next I will talk a little bit about the structural diversities of this, the expression products of these transporters. We just say that in this family it contains 10 genes, 10 different genes, 10 different members of transporters. At least these 8 are established, well-established bicarbonate transporters. And so there's huge diversities in the genes encoding these bicarbonate transporters. And on the other hand, each of these genes are able to produce a profound of diversities in the expression products of the these genes. Each of these genes contains at least 2 different promoters, different promoters, and they contain many several other cassette exons that can be alternatively spliced. For example, this is SLC4A10, which encoding MCN2. It contains 3 promoters and has 1, 2, 3, 4, 5, 6 gene exons that can be alternatively spliced. So if the gene is transcribed from the first promoter P1, then it will escape exon 2, 3, 4, and so contains exon 1, 5, 6, 7, 8, and so on. And then it will make a protein, MBCN2 protein, starting with MEIK. That's the first 4 residues we used to stand for this variants. And if it transcribed from the third promoter P3, it will contain exons 4, 5, 6, 7, 8, and so on. And it will, if the gene was transcribed from promoter P2, we get several different types of transcripts. Some of these encoding these three guys encoding the protein starting with MCDL. It has long entom, and if escape exon 3 or 3 and 7, then it will abort the open reading frame and encode the protein starting with MHA, and we call MPCN2. So so far we have identified about 16 different variants expressed by this single gene. These 6 guys have an N-terminal starting with MEIK, we call the MIC MPCN2. And those guys have an N-terminal, different N-terminal starting with MCDL, and those guys are truncated in the N-terminal domain, starting with MHAM. Okay. And also we found that different MPCN2 variants with different N-terminal has a distinguished expression profile in different tissues. For example, the MIC MPCN2 is highly expressed in the central nervous system, but this MCDL MPCN2 is highly expressed in the kidney. So this shows that the expression of different promoters of SLC4ATM is highly tissue specific. And this is another example of MBC of SLC4A7, which encodes sodium bicarbonate co-transfer MBCN1. It has 2 promoters. We have demonstrated that this gene contains 2 promoters and have several other cassette exons can be alternatively spliced. And so far we have identified about 20 different variants that have different structures. Basically this MBCN1 have 4 different N-terminals, starting with MEF, MERF, MEPRO, MDEL, and MID. And have different, some optional cassettes can be present or absence in different variants. Okay. we used synapse oocytes as the expression system to investigate the effects of these optional cassettes on the activity of these transporters. This is the system we just introduced improved slides, and we expressed the different variants in oocytes and compared the activities of different transporters. This is the apparent activity of different variants, and we also examined the surface protein abundance of these transporters by using a technique of biotination, and finally we got the if we divide this is the activity of the protein-dependent activity, and this is the relative surface abundance. If we divide activity with this surface protein abundance, we got an index of single molecular activity of these transporters. After sophisticated statistical analysis, we found that several cassettes, is the optional cassettes, cause 2, 4, and 3, have very strong stimulative effect on the activity of these transporters. Okay. How much time? And finally, I will talk about a little bit about progress on the project on the role of NCPTs in renal physiology. In our daily diet, we actually take more acids than base into our body. So we have to maintain acid-base balance, the systemic acid-base balance. We have to secrete acids and recycling base in our body. 2 organs are very important in our body, and one is lung. We exhale CO2, carbon dioxide by respiration, and then on the other hand, the kidney is the other key organ in our body. The kidney is involved in 2 key processes in acid-base regulation. One is bicarbonate reabsorption, and the other one is ammonia excretion. This is the diagram of the red nephron. You can see how bicarbonate is recovered. In the glomerulus, 100% of blood bicarbonate is filtered into the glomerulus and moved into different segments of the renal tubules. Then all this bicarbonate in the filtrate will be recovered in different segments. About 80% of the filtered bicarbonate is reabsorbed by the proximal tubule, this part. And about 10% is reabsorbed by the medullary thick ascending limb, and 6% here, and about 4% is recovered by the collecting duct. So then, virtually in the urea, virtually there's no bicarbonate is retained in the urea. If we have a look at the proximal tubular epithelial cell here, it shows the molecular mechanism of bicarbonate reabsorption by proximal tubular epithelial cells. Here, currently, the consensus is that the bicarbonate in the lumen of the tubule will combine the proton to form carbonate, which will dissociate into CO2 and water molecules. CO2 will flux into the epithelial cell across the apical membrane here, and then in the epithelial cells, the CO2 will combine with the water molecule to form carbonate, and then dissociate into proton and bicarbonate. The proton will be extruded by this proton pump or either sodium hydrogen exchanger, and the bicarbonate will be recovered by sodium electrogenic sodium bicarbonate cotransporter MBC-E1 here. So currently the literature study, a lot of studies have indicated that these 2 proteins, NHE3 and the proton pump, are responsible for about 80% of bicarbonate reabsorption in proximal tubule cells, and the 20% is accomplished by other pathways. And we found that, for the first time, there is a bicarbonate transporter expressed on the apical membrane here. And so our study shows it is likely that this protein's bicarbonate transporter is involved in bicarbonate reabsorption in proximal tubule cells here. And this is the first time to show that this bicarbonate transporter is expressed on the apical membrane. And I will show you data if I have time a little bit later here. I will just show you the diagram first. So this is the bicarbonate reabsorption in proximal tubule. And so this MBC-E1, so this process is very important to maintain the systemic acid-base balance in our body. If this transporter MBC-E1 is disturbed or mutated, then it will cause severe proximal tubule acidosis. So we have a lot of studies have identified series of mutations associated with acidosis, proximal tubule acidosis. Okay. So what I just showed is the bicarbonate reabsorption, and what happens to ammonium secretion. And in our kidney, ammonium secretion, ammonium is generated by amino acids metabolism in epithelial cells. Basically, it's by the metabolism of glutamine. The metabolism of glutamine will make ammonium and produce some bicarbonate. So the bicarbonate will be reabsorbed by MBC-E1 here, and this ammonium will secrete into lumen of proximal tubule. So when the ammonium is secreted into lumen of the proximal tubule cells, tubules, and then will travel through along the tubules here, and it travels down to this particular structure we call Henry's loop. And here the ammonium will be uptaken by these epithelial cells, and then secreted into interstitial space, and then finally get into the collecting duct here. And so there's very little ammonium that will be goes to this segment here. So the ammonium concentration will increase from the cortex to the medulla of the kidney. Okay, and so this process here, this ammonium transport across membrane, membrane here in these segments we call medullary thick ascending limb. If you have a detailed look at this segment here, the ammonium ions in the lumen will be uptaken into a epithelial cell by these transporters or even the channels. So, and then will be the ammonium uptake in the cell will cause a pH challenge to the cell, because this is acid. So we cause a profound acid pH challenge to the cell. So this pH challenge can be neutralized or attenuated by the uptake of bicarbonate at the basolateral membrane. Others have demonstrated that MBC-N1 mediates bicarbonate uptake to titrate ammonium fluxing. Okay, we found that MBC-N2 is also expressed at the basolateral membrane of these mTOR epithelial cells, and mediate bicarbonate uptake here to neutralize ammonium. Okay. So these are some latest progress in our lab and about MBC-N2 in the acid-base transport in renal. I think probably it's time to. I'm not going to show I'm not going to show the details of the data. And so basically, I just will go through very quickly. And I've just shown that there's some there's 3 different types of MPCN2 variants, right? There's MYC MPCN2 and MCDL MPCN2, and we found that different MPCN2 variants have distinct distribution in different tissue of the kidney. MCDL is primarily expressed in cortex and automedulla, where MYC MPCN2 is expressed in the medulla. And the MCDL MPCN2 is expressed in the apical membrane. This shows this is the immunohistochemistry staining. And MYC MPCN2, this the MPCN2 different variants with different N-termini are localized at the basolateral membrane of mTOR cells. And we show the effect of acidosis, ammonia-induced metabolic acidosis on the expression abundance of different variants of MPCN2. It has no effect on MCDL, MCDL, but has a huge, substantial increase on this MYC MPCN2. Okay, I think I'll just stop here. And so the summary is the NCPTs of SLC4 family are of a major group of pH regulators, and they are widely expressed in different tissues, playing vital, physiological, and pathological roles. And of course, these transporters can be potential drug targets, and they are associated with a lot of disease. And the 5 NCPT genes can produce a great extent of diversities by using alternative splicing, alternative transcription, use different promoters. And the expression of different variants are highly tissue-specific and cell type-specific. And the optional structural elements have important roles in the functional regulation of these transporters. And I would like to thank members in my lab, Dr. Yin Liu and Duncan Wans. This is a PhD student. He will expect his PhD degree in this summer. And it's the PhD students Li-Ming, Xu Zhou, is a master students have. And it's all master students. And also, I would like to thank my collaborators at Case Western Reserve University, Dr. Boren, which was my former mentor of PhD and postdoc training, and Dr. Chen, and Nathan Morris, and Musa Aziz Haifi. And also, I'd like to thank my collaborators in State University of New York at Buffalo, and Mark Parker, and so on. And thank you very much.
Thank you for your presentation. Only one question I'm going to ask. Those scenarios is in normal cells, right? So what could be a possible scenario in tumor cells or cancer cells to this transporter?
I'm sorry, so your question is...
what happens to this transporter in tumor cells or cancer cells?
Tumor cells?
Yes.
Cancer?
Cancer cells.
So, right, these transporters are widely expressed in not a wide range of normal tissues and cells. So, and I think we, in some, we know actually in cancer, cancer is I think it's in cancer it's a hypoxial environment, and cancer cells has it's very active in metabolism, but it's not so effective in the circulation. So this accumulation of acids in tumor cells. So I think in tumor cells there's a challenge of decreasing in the pH, and so to maintain the normal survival of cells, I think the cancer cells have to develop a mechanism to extrude acids, and also probably some mechanism to tolerate this in acidic environment. And we do know this some transporters that is involved in tumor development, and for example, as NBC N1 of this member NBC N1. is associated with breast cancer. I think there's some other aspartate transport, as I know, like sodium hydrogen exchanges, NHE, is involved in other kind of cancers. And there's also some proteins related to pH regulations. For example, carbonic anhydrase, that's an enzyme to catalyze the reaction of CO2 to bicarbonate. And those enzymes are also involved in probably in the pathology of cancers.
Yeah. Okay, thank you.